Okay, so once we've described the physical structure of a community, where it stops and starts, and whether it's gradual or distinct in its boundaries, the next thing that we need to talk about is who lives there. Because in fact, that is what community ecology is all about, is which species live there. And the way that we describe communities is by the number of species and their relative abundance within those communities. This is where I've spent a lot of my time when I've acted as a community ecologist, counting and categorizing species. And we use a whole set of metrics that describe how rich a community is in its species diversity. So we'll talk about that next.
So as I've mentioned many times by now in the class, ecology is an incredibly quantitative science. And one of the ways that we capture community qualities is we describe how many species are in a community and how common each of the individual species are within that community. And we call these species diversity metrics. So they're ways of measuring species diversity within those communities. So species richness is a very simple metric which is calculated as the number of species in a community. So you just go through a community, you figure out which organisms live there, and then it's just the number of species in that community that makes up the metric species richness. The thing is that we could have some organisms that are a few organisms that are very common, like an invasive species like garlic mustard. We could have a forest full of garlic mustard and one individual of 20 other species, and we would have the species richness in that forest of 21. But that wouldn't reflect the bias towards that one invasive species, the garlic mustard. And so another important metric that we use to characterize communities is the relative abundance of each species. And that is reflected in the proportion of individuals in a community represented by a species. So are there thousands of garlic mustard and out of 1,001 individuals within a community? Maybe there's 1,000 garlic mustard and one maple tree. So then you would see that the relative abundance of that community is really skewed towards one species. And then both species richness and relative abundance are incorporated to calculate another metric called species evenness. And this is basically a comparison of the relative abundance of each species within that community. So on the right-hand side, you can see a picture of someone or a graph of someone, R.H. Whitaker and Preston, two very important ecologists, old dead white guys to be clear, but very important ecologists who basically coined some of these species diversity metrics or started using them in real ways. And what they did was, for instance, they looked at the abundance of individuals of a species, right, on the x-axis, and then they looked at the number of species in that community. So Preston looked at birds, and Whitaker looked at plants. And so you can see that some organisms that are very common, there are only very few species that are very common in this particular community. And there are quite a few species that have an intermediate amount of abundance, and then a few species that are very rare in abundance. Okay, so that's what that's saying. But what's interesting is that this pattern happens across communities. And so we get this typical bell-shaped curve or normal distribution of how abundance relates to number of species. And this curve, this normal distribution or bell-shaped distribution, comes out when we use a logarithmic scale on the x-axis, which is important to notice. Now, the next thing that we do is we look at abundance. And I'm going to basically we can create these abundance curves to describe relative abundance of each species in a community in rank order, from the most abundant species to the least abundant species. So here you can see in figure A, we've ranked abundance. So this organism is the most abundant and this organism is the least abundant in two communities. We're looking at this wacky little type of lizard. And you can see that in community A, this lizard is the most abundant. And in community, and there are different species. There are different species that rank as second most abundant, third most abundant, etc. Okay. In contrast, in community B, we see there's no difference in the relative abundance of 5 species, which is kind of weird, really kind of weird. And that's because we didn't know what kind of communities we were looking at. But community A is actually a primary forest, and community B is actually eucalyptus plantation. So in this case, in the first image, all we're showing you is an example. But in the second example on the right, we're actually showing you real data. And so they've taken a relative abundance of lizard species, all different kinds of lizard species, not just this one. And you can see that in the eucalyptus plantation, there are literally only 5 species, or maybe 6, I guess, 5 or 6 species in the whole plantation. In contrast, in the primary forest, there are 16 species. They're ranked out of 16. But what's interesting is that 2 are very abundant in both types of environments. Maybe 3 are very common. But in the eucalyptus plantation, actually, all 5 are relatively abundant. In contrast, in the primary forest, we actually see quite a few organisms that are relatively rare within that primary forest. So this different environment allows for different kinds of diversity to appear, which is kind of cool.
Hi, everyone. All right, so now we want to talk about what are the influences on species diversity, and we're going to talk about that all week, really. I don't know if you can tell, I'm in Kings Mill Park, which is on the west side of Toronto, and right above me there is a male red-winged blackbird starting to sing, so spring is totally here. Keep on going with these lectures. I just want to send you all the positive vibes. But so when we start looking at species diversity, there's a number of factors that actually affect species diversity. That can include how much food is available, how many resources are available, how much space is available, what kind of diversity of habitats do they have to live in, as well as how much disturbance occurs within an environment and the type of species that are there. And specifically, we're talking about keystone species in here. We'll talk all about these in this next module.
In order to actually calculate measures of species diversity, you need to be able to do a couple of things, but mostly you need to be able to count and you need to be able to divide. So not really hard skills in math. So when we're calculating species richness, which here is described as S, all you're simply doing is counting the number of species within that community that have an abundance over 1. So you can see here that there are 5 species, mouse, chipmunk, squirrel, shrew, and vole, that all have abundances above 0, and so the species richness is 5. In contrast, over in community C, where we have only 4 species, voles are absent, you can see species richness is 4. And then in order to calculate relative abundance, what we do is figure out the total number of individuals within a population, within a community, as well as the number of individuals within a species in a community, so in each population. And we calculate their relative abundance as their abundance divided by the community total abundance. So it would be 24 divided by 100 is equal to 0.24, where 100 comes from the total abundance, which is simply the sum of all individuals of all species within a community. So there you can see the relative abundance of each species within a community, and this varies between communities. So you can see in community A, mice are relatively more abundant in community A than in community B, whereas in community A, squirrels are relatively rare, and in community B, they're as common as everyone else. So this just gives you an idea of how to calculate very basic metrics that describe species diversity within communities. Now, what's interesting is that species richness is often not random in its patterns, but rather species richness often varies with the amount of primary productivity or growth that occurs within a community. So primary productivity, what we're meaning there is how much green stuff is produced annually in a season. So literally grams per unit time. So on the x-axis, we can describe productivity. So this is a more productive environment, and this is a less productive environment. So deserts or Arctic might be down here, a waste mine, tailings area, and many areas like grasslands are highly productive, relatively new, but like forests, not super new, not just planted, but medium age forests are very high productivity. So that gives you an idea of what we're talking about in terms of growing green stuff. And then what we will see is that there are 5 different patterns of species richness, where high means that there's lots of species richness, and low means there's low amounts of species richness or close to no species richness. So we see very different patterns, and what's interesting is it depends on the organism that you're looking at as to what relationship you're likely to see. But generally, most organisms, whether it's aquatic vertebrates, so like whales or sea otters or dolphins or fish, we often see in communities of aquatic vertebrates, we often see a hump-shaped distribution. That is, in intermediate productivity environments, we see the highest species diversity. So intermediate productivity environments include things like old growth forests, well-established communities that may not produce as much green stuff as medium age forests, but have an enormous amount of biodiversity within them. So, but within aquatic vertebrate communities, the kind of medium productivity environments, we'll see places around the world, and we'll show you some biodiversity hotspots. They tend to not be in those extremely high productivity areas. I shared some pictures of those in our first week of lectures. They won't be there. That will be slightly lower species diversity, but they'll be off coast, not every coast. They'll be in particular regions like coral reefs. Those are the sort of spaces that we might see them. The same with aquatic invertebrates. We also see the most diversity, the highest productivity and its relationship to species richness is often described by a hump-shaped pattern in aquatic invertebrates as well. Same with aquatic plants. It's kind of fascinating. There are a non-negligible amount of studies, however, that also show no relationship to productivity and species richness. So you will see this light blue line is also a common pattern where sometimes productivity does not predict species richness. So it's not the only way we will see high species richness is intermediate productivity. Sometimes it's not related at all. In terrestrial vertebrates, we either see hump-shaped patterns or positive relationships. So in terrestrial environments, so this will be in the positive relationships. What we're talking about is under high productivity environments, we'll see high species richness, and under low productivity environments, we'll see low species richness for terrestrial vertebrates. For terrestrial invertebrates, the hump-shaped pattern overwhelmingly predicts the patterns between species richness and productivity, or the relationship between species richness and productivity. And we also see the same pattern in terrestrial plants, with some studies finding positive relationships similar to terrestrial vertebrates. It's interesting, right? But this would give us some information that often, if you save medium to high productivity environments, you're going to be protecting a lot of species richness. Now, that's not to say that the opposite can't be true. There are cases where we see negative relationships between productivity and species richness, or U-shaped relationships between species richness and productivity, but generally, the most common patterns are to see species richness and productivity being represented most commonly in the positive and hump-shaped patterns for most organisms, for most vertebrate and invertebrate organisms, as well as plants. Remember, of course, that this does not describe any microbial patterns, which is interesting to notice, right? All of that information is absent. Now, why is this the case? People have been asking this question of why does primary productivity influence species richness? And one of the answers that people came up with relatively recently was looking at how resources available in those locations might influence primary productivity and therefore influence species richness. So what I'm showing you is a picture of a grassland. It's a grassland that's actually an experimental grassland. And a researcher in the US named David Tillman has been performing research on this space for a very long time. And essentially what he's followed are grassland species, which are relatively high productivity, and how those grassland communities change in response to fertility. So what he did was he created plots with same species within each of them planted, and then he fertilized them in different ways. And you can see, even just looking at what you're seeing in front of you, you can see different amounts of vegetation, different colors of colors of vegetation, which suggests either different species composition or different chlorophyll contents at least. And this is fascinating because he was then able, with a lot of student researchers, he would go in with his team and look at which species were present every year. He did this for many, many years, and there's a massive amount of publications coming out of David Tillman's lab. But what the general message is from his lab, and this was published in Nature in 2007, was that if you added more fertility whether that's nitrogen, phosphorus, potassium, all of the micronutrients, however many resources you added to the plot, you would actually see a decline in species diversity. And what he was finding over and over and over again was that the more resources you add, the more likely there was going to be one dominant organism that would take over the plot. That one dominant organism was a better competitor and scavenged more of the resources so that it excluded the others. Now you see why we were talking about competition last week, right? It's kind of fascinating how these interactions between species then leads to consequences for community level species diversity. So people have been thinking about how species diversity has been affected by their environment for a very long time. And in this case, I'm showing you an example from Robert MacArthur, one of the preeminent biodiversity ecologists in ecology. He's certainly well revered in terms of his contributions to our field. He did a study in 1961 where he went and looked at different forest patches. And in the forest, he asked, were all the trees at the same height? So were they all at the same height? Or was there uniformity, which would be here? Or was there lots and lots and lots of diversity? So this is, I said something wrong here. At this point right here, they weren't all uniform. This is where they would have been uniform. Here is where there's about, equal amounts of every kind of shrub. And this would be more variation than you'd expect by chance alone. And so what he saw, so you can imagine some trees, at least at low diversity, you might expect you're in a plantation and every tree is the same height, whatever the height is. And so birds can only perch or sit at one particular height, right? In contrast, when we're looking at something that a forest that is very diverse, we might see some that are shrubs, some that are giant trees, some that are intermediate. We might see lots of diversity of tree size in this case, all right? Those are terrible tree drawings. I'm just drawing with a mouse, which is, hard. And what he found was that the more diversity in height that you have in a forest, the more diverse bird species communities you will have. The more uniform a height, and so I'm thinking of plantations, or I'm thinking of, say, a grassland where everything is almost identical in height. In those cases, you're going to have relatively low diversity. Now, grasslands actually have quite high diversity, so I suspect actually he was only looking at forests, and this might have been a plantation of some very specific sort, because grassland's actually pretty diverse. But forests will have one uniform height, and that's all he would have seen, is very few species. Now, of course, when he's talking about bird species diversity, this is not species richness. This is another metric of bird species diversity, just to be clear. So it wasn't that he was seeing one species or two species or three species. This is likely on a log base 10 scale. So this is 10 species, 100 species, 1,000 species, right? So I suspect that's what actually was going on there. All right, so another way that communities are influenced or form is around a keystone species. And before I go into what a keystone species is, I want to show you what a keystone is. So keystones exist as an architectural element in arches. And the keystone is the last stone placed. It's the stone at the top. And its placement means that every other stone will be held in place, and you don't actually need mortar. But we add mortar to make it so that no one can knock out something and then everything falls down. So the keystone is actually the element that holds the entire wall together. Similarly, a keystone species is a species that substantially affects the structure of communities, despite the fact that individuals of the species might not be particularly numerous. You can see there's only one keystone here. You don't need many of them to hold the whole environment, the whole wall together. Same with a community. So what do keystone species in a community look like? Well, here's an example of one keystone species. So sea stars, starfish, right? These purple guys are considered a keystone species. And what is the argument for that? Well, when a scientist studied the presence or absence of this keystone predator, it consumes all of these other mussels, sucks them right out with its mouth part that's on its belly. What you could see was that when they were present, there was a maintenance of high species diversity within that coastal community. In contrast, if the researchers went to a second community and removed all the sea stars from that community and kept visiting to make sure there were no sea stars returning, what they noticed was a radical drop in the number of mollusk species present in that environment. So the researcher didn't destroy any mollusk species. It's just simply that the keystone predator, that sea star, was actually maintaining dynamics between the mollusk species so as to maintain high species diversity. So often conservation programs manage a single keystone species. They focus all their efforts on one species and making sure it's conserved, to hope that that will help conserve a whole host of other species in the environment without having to do anything. It's a relatively inexpensive way to approach conservation that's often very successful. The last kind of organism that I think is important to point out is an ecosystem engineer. Now, ecosystem engineers in some ways create communities. So a beaver, in its efforts to create an environment that allows it to have a safe home, will chop down trees, right? So it chops down those trees. It's an amazing, amazing animal. It will chop down very large trees. Often, I don't know if you can see this, but this is poplar trees, loves poplar trees. It will chop them down. It harvests the small branches and stores them underwater for feeding during the winter. For the large branches, it uses those large branches to dam rivers or streams. So it will prevent the water from moving as quickly down the river bed or the riverbed, creating a pond. So this, what we imagine actually happened was there was a stream that was this wide. And now look at what this beaver has created, this pond. It's actually 4 feet deeper or so, maybe 6 feet deeper than it was, and it's massive. So all of a sudden, this beaver has a place to put its den. So here's its den right back here. It will have a mud hut that it can swim out of. It never needs to go onto land to get out of its home. It can exit through the water and swim through there. It stores these small branches in the water for overwintering food supplies. And it has entirely changed this environment, because all of a sudden the riverbanks are not here, right? I'm guessing that it went over this way. But rather there's all of this that is flooded, all of this that is flooded. Now we can have organisms that live in deep water live here compared to running deep water and still water, this is a relatively slow moving body of water now, this organism has entirely changed the ecosystem that's there. By doing so, this beaver has introduced new communities of organisms, not by bringing them, just by actually making their an environment capable of supporting those organisms. So ecosystem engineers have radical impacts on environments and can create new communities by their very presence. So the beaver created quite a disturbance there, created a whole new natural environment, and in fact has probably boosted species diversity in that particular region. And that starts humans asking the same question. So why is it that the beaver can disturb an environment, create a new home, and everything is hunky-dory? In fact, everything is excellent. But when I, as a human, want to go and build a new home, then I get in trouble because I create a disturbance and nothing comes and lives there. So ecologists have been thinking about this for a very long time and have put together a hypothesis to describe why the beaver can do it and we can't. And it's called the intermediate disturbance hypothesis. The hypothesis goes like this. When disturbance is very rare, then actually we're going to see relatively rare numbers of species because we'll only see those species that flourish in environments of that particular kind. But when we disturb, the moment we actually radically disturb, some organisms that were living there might still be surviving. They might not be thriving, but they might be surviving. And other organisms that couldn't compete come in, and we start to see an increase in species diversity. Now we see an increase in species diversity until some intermediate level of disturbance, where those individuals that can handle lots of disturbance have arrived, and those who can handle a little bit of disturbance haven't died. So we have this maximum number of species or maximum diversity right there. But then if the disturbance is so great or is so frequent, like humans walking on the same piece of grass over and over and over and over and over and over again, we're going to start to see that organisms die and nothing can survive that disturbance. And ultimately, we see a reduction in species diversity again. So ecologists propose that in a community that experiences some disturbance, a moderate amount of disturbance, they're going to have more species present than in communities that either experience frequent disturbance or even very rare disturbance. And here's an example where ecologists were looking at snail density. Now, snails are a herbivore, they're a grazing herbivore, so they actually cause disturbance wherever they go. And if you put one into your aquarium, they will keep it nice and tidy, free of algae, right? Because that's what they're grazing on, is all that algae. So what they did was they looked at how species richness of algae in a particular community varied with the number of snails present. And again, in communities with very few snails, then there was actually relatively low algae diversity. In communities with high snail density, there was also relatively low species diversity of algae because they ate it all. But in intermediate amounts of snail diversity, snail density, they actually saw the most species richness of algae, which is kind of cool. All right, so that is our conversation on species diversity in communities. I'm going to move on to the next module.